Thank you. Well, civilized people aren't even out of bed yet, so I'm not sure that I can really stand too many mornings at 8 o'clock. But anyway, for those of you who have come, thank you for coming and making my getting up so early worthwhile. And, I thought today being election day was going to be a vacation anyway. Still, today we're going to continue on in the first half of this lecture with our pursuit of the fascinating class of viruses called retroviruses. And as I indicated to you yesterday, once we've seen how retroviruses cause tumors, then we'll come back to the DNA viruses and see how they cause tumors as well. And yesterday we left off at this point here, looking at the typical retrovirus. And these are really quite small viruses. A genome of about 10,000 base pairs. Not much you can encode in 10,000 base pairs. But there is one gene for the internal proteins, one gene for the enzymes, and one gene for the proteins on the surface. And these are envelope viruses that bud out through the surface of the cell. So this is a typical retrovirus. It's all you need to make a retrovirus, a new progeny virion. Now, we saw yesterday that when these are integrated into the DNA, they are slightly different in that this U3 gets transposed over here and the U5 gets transposed over there so that the genome can be copied using RNA polymerase II into a new RNA genome, and that gets over the problem faced by this virus of the fact that RNA polymerase II does not copy promoters, initiation sites, and all that kind of stuff that is necessary for the functioning of a gene. So this is all that's necessary if you're a retrovirus. And a retrovirus like this may or may not cause cancer, but normally this probably would not cause cancer. So what makes other retroviruses actually cause cancer? Well, the first retrovirus that was shown to cause cancer was Rous sarcoma virus. And this causes a tumor that is found in chickens and goes through rather rapidly flocks of chickens. And when people looked at the structure of Rous sarcoma virus, they found what they expected here, a GAG, a pollen and env gene, but they found something extra. And this was called the Sarc gene because this was the gene that was rapidly identified as being responsible for these organisms, these viruses, to cause the tumor in the chickens. If you deleted this gene, you've still got progeny virions, but you could not form a tumor. So this fitted the definition then of what we talked about yesterday of the oncogene. So this is an oncogenic sequence that is extra to what is necessary to make a retrovirus. Now, contrast that with those oncogenic sequences that we saw in the DNA tumor viruses, which were the early functions. And these early functions change the DNA metabolism of the cell that's been invaded by the virus. And these are necessary not only to cause tumors, but also for the proper replication of the DNA virus into progeny virions. So here's the difference between the oncogenes of retroviruses, RNA tumor viruses, and the oncogenes of DNA tumor viruses. The DNA tumor virus oncogene is a functional and essential gene of the virus. This is an extra gene, which in fact, as we shall see in a few moments, has been picked up during the life on Earth of this virus from host cells. Now, that was the first of the retroviruses that were investigated, and the structure was elucidated. Others were then followed. And it became quite clear that you really don't need that GAG and POL in many of these tumorigenic viruses. For example, here, avian myeloblastosis virus has lost its env gene and substituted it with an oncogene, which was called MIB, and you can see they're usually referred to as three-letter code. And here's feline sarcoma virus, and it has lost virtually everything that codes for retrovirus, but is substituted with FMS, which is another of these oncogenes. Now, of course, these here cannot form a new retrovirus unless they're in a cell that's co-infected with a helper virus that will provide the polymerase, that will provide the internal antigens, and will provide the envelope glycoproteins. But provided they are co-infected with a functional retrovirus, which has all three genes, they can take those proteins, incorporate it into the virus with these genomes, and then spread to another cell. But to form tumors, we've got these oncogenic sequences here that can be replicated in the cell because they do have the terminal repeats that allow them to integrate into the genome of the organism, the host cell that's been invaded, and therefore to replicate their genome into more sequences of RNA. And of course, when that's transcribed to make them more RNA, they also transcribe these genes here. So these genes then, here in purple, are the genes that are responsible for the tumorigenic properties of these retroviruses. They are something that has been picked up by the virus during evolution and allows them to have this tumorigenic potential. And so we call them as oncogenes. And normally we call these extra genes viral oncogenes, or we shorten that to VONC. Now, having discovered that, this happened in the 1980s. And in the 1980s, techniques of DNA hybridization were being developed by scientists who were interested in the structure and function of viruses. And these hybridization techniques allowed people to ask a simple question. This viral oncogene is here that is being extra. It is extra to what the virus needs to make its 3 primary protein products. Where did it come from? Perhaps it, in fact, came from, as I mentioned earlier, cells through which the virus passed in the past. And so people started looking, are there any homologs of the viral oncogenes of RNA tumor viruses found in normal cells? And you could do this by DNA hybridization studies. And to everybody's surprise, they found out, yes, there are. For every one of these retrovirus-carried oncogenic sequences, you and I have a cellular homologue. And it looks what happened in Rous sarcoma virus is that during the development, the evolution of this virus, at some stage, it picked up a cellular gene which was then put into a new context and mutated, because remember, these viruses have an enormous mutagenic potential. And in mutating, it went out of control and allowed the virus then, in the infected cell, to turn on cell division and cause a tumor. But clearly, if you and I have all of these cellular homologs, we're not growing all the time. So in us, these cellular homologs are not switched on all the time. They're not causing our cells to divide all of the time. But they were switched on during your development, when your cells divide in particular circumstances, when you develop from a gleam in your mother's eye up into what you are now. At some stages in your development, these genes were essential and had to be expressed. But during the passage of the virus through the host organism, it's picked up these genes, mutated them, and then uncontrolled cellular replication has followed. So there are these proto-oncogenes, about 30 or 40 of them, that we have that have been picked up by retroviruses and put to the nefarious ends of the retrovirus, which is to produce more retroviruses, which is the nefarious end of any virus. So proto-oncogene then is simply a cellular host gene that is homologous with a similar gene in a transforming virus. And this cellular oncogene only induces tumors, presumably, after mutation or some other change in the genome. So it was the discovery then of these acutely transforming retroviruses like Rous sarcoma virus and their accompanying oncogenes that explained how these viruses could cause their host cell to replicate its DNA endlessly and form a large number of progeny tumor cells. Okay? And these are known as the acutely transforming retroviruses. If you infect a lab animal with one of these things, it gets a tumor very, very rapidly. It expresses a protein in the host cell that turns on cellular DNA replication, cell division. Okay. But then a class of retroviruses was discovered, and these retroviruses are the chronically transforming retroviruses, which do not cause tumors rapidly. They may cause tumors in an animal situation over a period of years. So, the next question then was, why do these cause tumors? And when people looked at the structure of these tumor viruses, these retroviruses, they had a surprise. Here's avian leukosis virus, and what you can see is it has no extra gene. And yet we've said in the case of RSV that it needs to have that extra Sark gene in order to cause a tumor. And it does it very rapidly. But these guys here appear to be able to cause tumors. They don't have an oncogene. There's nothing here that differentiates this from a non-tumor causing retrovirus. So how do they cause tumors? No oncogene. Well, the clue to that came from the studies of where this virus, avian leukosis virus, integrates into the host genome. Now, retroviruses can integrate into your genome, into my genome, into a chicken's genome, all over the place. They don't really care where they integrate. They just want to get in there so they can duplicate their DNA and make more RNA. But when you look at the tumor, you find that in the tumor cells, the virus is integrated at one or a very limited number of places. So there must be something very special that when you get this virus integrated at that point in the genome, that then the cell becomes tumorigenic in the absence of a viral oncogene. And that suggests that all the tumors arose from one cell, where the virus got in at a particular place, i.e. that the tumor is clonal. And there must be something about this rare event that's very, very important. So people looked to see where it was. What was the context into which the virus inserted itself in order that it may turn the cell on to unlimited DNA replication and mitosis. So what was special about that site? Well, it turned out that all of these viruses had been inserted next to one of the cellular proto-oncogenes. In this case, c-Myc. So the virus is turning on these cells to unlimited replication, not by bringing in a viral oncogene, which tells the cell, start dividing. What it's doing is, it's taking those fantastically efficient promoters, which we know as the LTRs, because viral promoters tend to be rather efficient because they want to make lots and lots of copies of the virus, and inserting it just upstream of a cellular proto-oncogene that is normally not expressed except at times when you want to divide your cells during development. But in this case, that super promoter is being inserted and taking over the control of this rather benign gene that you need for your development, for your DNA replication, as you are growing up from being a fertilized cell into what you are now, and switching it on so that cells actually start to divide. And this is referred to as oncogenesis by promoter insertion. So this is putting the viral's very active LTR promoter upstream from a cell division control gene, taking it over, switching it on, and creating in the cell an environment for unlimited DNA replication, which of course is to the advantage of the virus, and then as a byproduct of that, forming many, many new cells when they shouldn't be formed, and therefore a tumor. So that's how these oncogenes appear to be able, the cellular oncogenes are also involved in viral-mediated oncogenesis. So the question then arose, can these cellular oncogenes be involved in non-viral tumors? And the answer is yes, they can. When they get mutated, we know that some of the tumors that the human population gets are the result, probably not of a viral infection, but mutation in these cellular homologs of viral oncogenes, these proto-oncogenes. As I said, they control DNA replication and cell division. Some of them are growth factors genes that you produce when cells are supposed to divide. But in the case when they're mutated, they send the signal all the time. Therefore, the cells divide all the time. Some of them are signaling proteins inside the cells that send signals from growth factor receptors, and indeed some of them are growth factor receptors. They become mutated. This one over here, this growth factor, cis, it's actually platelet-derived growth factor, but it's mutated so that it sends this super signal to the cells, and they grow very, very rapidly. Some of these are internal signaling molecules, kinases. Some of them are transcription factors, which signal transcription of DNA into more DNA, cellular duplication, or transcription of DNA into RNA. So there are these 40 or so proto-oncogenes, normal cellular genes, that are involved in normal growth, that can be switched on by putting a viral promoter in front of them to give abnormal growth, or if they become mutated in some way in the absence of a virus, can also switch on abnormal cellular growth. Okay? So yes, these proto-oncogenes can, under the right context, the right mutation, also cause tumors. So they're growth factors, growth factor receptors, signal transduction proteins, transcription factors. You need them, we all need them for normal development, but when they get switched on abnormally, too much cell division. So now we've seen how retroviruses do the job of turning on cells, either by carrying in an oncogene or by taking control of one of our proto-oncogenes. And now that can bring us back, that knowledge can bring us back to the DNA tumor viruses. And this is a picture of the karyotype of a human cell, and it's just up here to remind me, to tell you, that we know where all of these oncogenes are in the human chromosomes. For example, we've mentioned MYC, and MYC is over here on chromosome 8. Now, going back to the very first DNA tumor virus that was discovered, which is the virus that causes Burkitt's lymphoma, Epstein-Barr virus, when you look at patients who've got Burkitt's lymphoma, what you see is they've got breaks and rejoining of their chromosomes. So they have hybrid chromosomes. And a picture in another situation of acute myelocytic leukemia is shown over here. You see this one up here, which is indicated by the arrow. You'll notice it is half yellow and half blue. That's a chromosome that is broken in half at some stage as a result of the infection of this cell by a virus, probably. And as a result of that, part of the one chromosome has joined to another. Now, when you look at Burkitt's lymphoma patients, they all have a break. There are actually three genotypes of these things, three phenotypes as far as the chromosomes are concerned. They all have a break near this, in this chromosome 8 here. And commonly, you'll get a translocation from chromosome 8 over to chromosome 14. Chromosome 8, just at the point where the chromosome has been broken, there is the MYC gene, one of these approximately 40 proto-oncogenes involved in growth control. So why are these cells in Burkitt's lymphoma, this is a B-cell lymphoma, why are they growing so rapidly? Why are they dividing when they shouldn't be dividing? They carry this breakage here. All three types of Burkitt's lymphoma, of the endemic type of Burkitt's lymphoma, have a break at chromosome 8. Clearly what's important is what's over here at 14 that's getting joined on to the rest of this chromosome. And the answer is, in Burkitt's lymphoma, in a large number of cases, what's over there, when you've got an 8 to 14 translocation of the chromosome, is the Ig heavy chain gene. And these are B cells that are designed to make immunoglobulins. So this is a promoter that is very active in B cells. And so what's happening is that when you break the chromosome, you're putting the MYC gene, which is normally quiescent, since we're not growing all the time, next to this Ig heavy chain promoter. And the Ig heavy chain promoter, when the cell starts to need to express the immunoglobulin heavy chain, instead it produces MYC. And therefore the cells respond to the production of MYC by the production of DNA. Simple as that. Now, certain people who have Burkitt's lymphoma do not show this 8 to 14. They have an 8 to 2 translocation. Well, it turns out that what's happening here is you're bringing MYC up against the light chain promoter. And so when the cells need to make light chains, instead they turn on MYC and the cell divides. Isn't that neat? Now, some other lymphocytic leukemias, B-cell lymphocytic, another of these, BCL is another of these proto-oncogenes. That one is brought up by a translocation next to either the Ig heavy chain or the Ig light chain promoters. Now in T cells, much the same thing. You see translocations in T cells, and as a result, what you get is the T cell receptor being brought up against, again, one of these oncogenes. So, it is clear then that the way Burkitt's lymphoma or Epstein-Barr virus causes Burkitt's lymphoma, it also involves these proto-oncogenes that were discovered as being carried by retroviruses. In this case, the virus is causing, and we don't know how, the virus is causing these translocations And when that translocation, breaking of the chromosome and resealing of the chromosomes to a hybrid chromosome, occurs at the right spot, so that MYC is brought up against a very active promoter in the cell, then up comes the level of MYC. So that's really quite neat. Now, all of these proto-oncogenes, when they receive mutations, when they have mutations, they tend to be dominant mutations. They tend to be what are called gain-of-function mutations. However, as knowledge of oncogenic phenomena expanded, there seem to be exceptions to this, and certain other oncogenic genes are recessive when they're mutated. And let's see if we can just... These are called anti-oncogenes. These seem to be opposite way around. These are tumor suppressor genes. Okay? So they're loss-of-function mutations, and the two best known of these are retinoblastoma RB gene and P53. And P53 is involved with RB in an enormous number of human cancers, whether they're virally induced or carcinogen-induced or whatever. So here's what's happening when you have a proto-oncogene. This is the things like SARC and MIB. Say it's a transcription factor that goes in and signals, as MYC does, that the cell should divide its DNA. It switches on the transcription of DNA into DNA. Okay? So, under the normal circumstances, this product here would bind to some site in the DNA, and it would signal, under the right circumstances, to duplicate that DNA. However, if you've got a mutant allele that always binds, just one of the alleles in the heterozygote that always binds, then you'll have a dominant mutation. Because this guy here, irrespective of the fact that this guy here will only bind under certain circumstances, he will always bind, this purple one. And so he will switch on, even in the heterozygote, the duplication of DNA. And of course, in the homozygote, you will have two alleles here, both of which make a mutant protein, which binds to these sites in the DNA and switches on duplication of DNA. Okay? So these type of things, like the proto-oncogenes here, when they get mutated, they tend to be dominant mutations. So they always bind like that. Now, in contrast to this is something like RB gene. Now, RB and P53 are tumor suppressor genes. When they are expressed, unlike the oncogenes, when they're expressed, they cause cells to divide, to duplicate their DNA. In the case of these genes, when they express their protein, it actually suppresses cell division. And there are certain circumstances where you want to suppress cell division. So the RB gene here forms the RB protein, and it will normally bind and turn off DNA replication. And even if you've got an RB protein here that doesn't bind because it's mutant, you've still got the functional protein from this allele, and therefore you will still get the wild-type situation of control of DNA replication. But in the mutant, when you've got two non-functional RB proteins being produced from two mutant genes in the homozygous over here, then there'll be nothing to bind onto the DNA and stop cellular DNA replication, and so as a result, you will get growth. And the retinoblastoma gene has been found to be involved in quite a few cancers, and the P53 gene also in quite a few. P53 is often inactivated in people with cancers by deletions and point mutations. And this now brings us back where we started yesterday. Because yesterday we talked about DNA tumor viruses and the fact that the way they cause tumors is to express these early genes that are involved in priming the cell to become a factory for more viruses by switching the cell onto an environment that is conducive to DNA replication, which is exactly what our virus wants. Okay? And we said that there was these early control regions. We didn't have to express any of the structural genes in the transformed cell. Indeed, we could have mutant viruses that have no genes for the structural proteins. But when we put these early genes in, E1A region we mentioned yesterday, you don't have to remember these terms, but these regions, these early regions of the genome, when we put them into the cell, when the virus takes them into the cell starts to divide because it is being put into a situation where DNA synthesis is being promoted. Now, all of these sequences here that are found upstream, as early proteins in adenoviruses, SV, polyoma, BK, all these other papillomas, they have sequences in them in these early regions that are very similar. They're sequences that will take over the replication control of DNA in the cell. Okay? And if you can abolish the oncogenic potential by mutating these regions or deleting these regions. So the proteins that are made here, like the E7 protein of papillomavirus, clearly are very, very important to the oncogenic potential of this virus. Now, when you look in, say, an adenovirus infected cell, normally in a non-infected cell, here's the RB gene, does its job, it's a 105,000 molecular weight protein, it's doing its job, it's sitting there, under most circumstances, it sits there on the DNA, on the chromosome, and says, this is not to be duplicated. So it stops duplication. When you look in an adenovirus infected cell, or even a cell infected just with this E1A region, this oncogene of the adenovirus, what do you see? Well, what you see is that the RB protein is actually complexed with the E1A. And by doing that, the RB protein can't bind to the sites where its job is to be done on the chromosomes, and therefore the cell says, I've got the signal to divide, and it does. In the case of some of these other ones, P53 does a very similar type of thing. In the case of hepatitis C, there's a protein made also that binds and inactivates P53. So one of your cell cycle control proteins, P53, your tumor suppressor genes, your tumor suppressor proteins, is knocked out by complexing with a protein made by hepatitis C. Now, in the case of papillomaviruses causing cervical cancer, etc., a protease is induced by the virus, specifically chops up P53. No P53, no cell cycle control, cell goes into DNA replication. So it was the discovery originally in the retroviruses of the oncogenes that gave rise to the idea of the proto-oncogenes and their involvement in both virally induced cancers and non-virally induced cancers, that then gave the rise to the idea of anti-oncogenes, DNA suppressor genes, and that happens to be the mechanism that has been adopted by most of the DNA tumor viruses in the way in which they cause cancer. Okay, so that takes us through, with our retroviruses and our DNA tumor viruses, to the end of oncogenic viruses, and I now would like to switch gears unless anybody wants to object or ask something.